am Luka and I was solving the problem 7, worms. The research question of my problem is earthworms change the mechanical properties of soil and make the soil more porous. Investigate this process and introduce quantitative parameters. I investigated this process by looking earthworms' effect on the porosity of the soil and how they affect growth of plants. So earthworms, they inhabit the sea, they inhabit the soil in the sea, and they inhabit the soil on the land. They live in sediment or in water ecosystems in the ground. They perform a very important ecological part in ventilation of the ground or soil. So I studied the common earthworm, Lumbricus terrestris. It processes large amounts of soil and organic matter mixed with soil in the digestive tract, and they create humus, which is important in the process of mineralization of organic matter. These activities enable better airness, texture, and soil structure. Other kinds of worms actually digest the anorganic matter, and they create humus from it. So there are other kinds of these worms, but I studied this one particular because it was the most common one in our area. So my hypothesis was that a larger amount of earthworms will create more pores and increase the soil porosity and the soil humidity. And the impact of earthworms on the plant growth will be suitable, and with more earthworms the plants will grow bigger and have bigger mass. My first experiment was I created 3 wooden boxes, and in each box it was clay soil mixed with organic matter 400 grams and humus 100 grams. The first box had no earthworms, the second one had 25 of them, and the third one had 50 of them. The temperature of the room was around 20 degrees, variation was 2 degrees Celsius. The soil had been moisturized every 48 hours with 500 milliliters of water to keep it humid so the earthworms wouldn't die. And the experiment lasted 28 days. So afterwards we took samples to determine the porosity and humidity of the soil using cylinder by Kapetsky. We calculated volume density. It is the ratio of mass of dry soil after drying at 105 degrees Celsius in a vacuum desiccator, and its volume in the natural states. The cylinder by Kapetsky was 100 centimeters cubic. And we calculated solid soil density. It is the mass of dry soil after drying on 105 degrees and its volume. And the porosity is calculated by this formula here. My second experiment had the same conditions as the first one, but we planted 100 seeds of radish in each of the boxes, and we measured the final mass on the plants to determine if earthworms had positive effect on the plants. So the starting mass was 0 grams, and we calculated the mass after 20 days. These are the results of the percentage of water in the soil. So the first box had 22.8 degrees moisture, second one had 24.47%, and the third one had 27.87%. So these 3 bars represent the first, red bar is first measuring from the first box, the blue one is the second measuring from the first box, and the third one is the third measuring from the first box. The same for the 3 boxes. And on the other axis is the percentage of water in the soil. So the first one, water evaporated faster because capillarity established, and in the second and third box, earthworm activity and their secretion of their slime disarranged establishment of capillarity, and water evaporated slower. The results for other experiments, I haven't got them because the researchers that helped me in this work are still working on them. I couldn't make them work faster because it's a long process of drying the soil and calculating the porosity. So I haven't got these results yet, but I expect the results to be similar to this one. So the porosity will grow with the number of earthworms, and the plant's mass will be higher with the number of earthworms. And that's it.
Okay, clarifying questions by the opponent.
Okay. Can you show me the first graph again?
Okay. Here it is.
Okay. Can you please explain what exactly does this graph show us?
So we see 3 boxes, 1, 2, and 3 numbers down below. From the first box, I took 3 samples of the ground with cylinder by Kopetsky, and I did the same with the second and third box. So I made 3 measurements to see if it was different on different parts of the ground. And as we see, it's pretty much the same. There is very, very low difference between the measurements. And the percentage of water grows with the number of earthworms. In the first box there is no of them, in the second one there is 25, in the third one there is 50 of them, so it grows gradually.
Why are there no differentiations?
Because the measurements are so similar, they are very well measured. Let's say I took from 3 different parts of the box, so if I got bad, let's say if I got 20, 25 and 30 from one box, that would be bad, but this is good because it's a similar percentage of water.
Okay. That's it. I don't have more.
Okay, so you have 3 minutes to prepare for your statement.
Okay, so time's up. You could finish, please.
So, statement by the Mikšić.
The program was actually composed very well, but I feel like the answer to it wasn't completely given. I feel like there could be a bit of an upgrade at the end of the program. Then yes. So in theory, a very good thing I noticed is that the reporter named the worms, and he had a picture that described the anatomic parts and organs of the worm, but he didn't explain how food is processed in the body of the worm, and how do these organs actually, how do they interact during the consummation of the soil. Then for the experiment, pro is that parameters were very well isolated, but there wasn't an analysis of soil before and after measuring. At least it wasn't mentioned. And for the end, the hypotheses were mentioned in the end, so that is satisfactory. But I think the conclusion could be done a bit more. I think there should be a bit more talking in the conclusion about other parts of the experiment. But it was all right. I have some questions about your experiment, and that is, why didn't you change? You said that you didn't change the conditions in the second experiment. What would happen if you did?
So everything I had, the conditions in the first experiment were optimal for the worms. I decided the temperature would be 20 degrees because they are most active at 20 degrees. I decided to water them every 2 days because I seen the soil being dried out after 2 days, and 500 milliliters seemed perfect because I had wooden boxes, so yeah, water could go through them. But I tested them before, so 1 liter of water would actually go through the wood and it would be too much. And half a liter of water was perfect because it didn't leak at all, and I chose wood and not some material that was completely not, the water wouldn't go through it, because the water would pile down at the bottom and it would just grow and it would kill the worms, because the worms actually breathe through their skin and they would drown practically.
What is actually the limit temperature where a worm can't function properly, can't process soil?
I don't know the highest, but I believe their optimal is between 15 and 25 degrees Celsius.
Okay, that's all right. How does porosity actually grow? In the experiment, you said that it does grow, but can you describe it a little bit more?
Well, I couldn't say it because I haven't got the results, but I believe that it would grow either exponentially or linear. I don't know which one of those would happen.
Okay, yes. But when you get the results, you'll
yes, I'll process them and I'll have more conclusion and everything.
Of course. And can this observation be described via mathematical formula?
Well, yes, I used the formula to get the results for porosity, but I don't know the results yet, so yes, they can be.
Okay. That is all.
Okay, there is 2 minutes to prepare review. For clarifying questions, the review. Marko, you should now ask questions.
Yes, but I don't have questions. I should write.
You don't need to write them, just ask. If you don't have any questions, we can continue to your statement. Okay.
Well, I think that problem is solved good. You don't have results, but it's not your fault. Your hypotheses were good, and I think you should try to experiment with other type of worms, and you should mention that how worms make better, and I think that the opponent was good, the questions were good, and
Okay, a statement by the opponent.
Okay. I actually like the execution of the experiment, and yes, the results aren't finished yet. But when they when they will be, the experiment will be finished, and I really hope that we can find a way to describe the observation a bit better. But other than that, I have no other remarks towards the review or the presentation. Experiment.
Okay, statement by the reporter.
I would say the reviewer did his job all right. He could ask a question, but he didn't. That is the only fault, the only wrong thing that he did. The opponent could ask more questions. He had some time left over, but his questions were all right. And I would like to thank them both for saying the things they did. That's about it.
Okay, now the questions of the jury. Garner, you go first.
How do you choose the size of the box in which you put the soil?
I chose some optimal size which seemed normal for the worms to have enough room to do the things they do.
Do you think that the size of the box could affect their activity?
If I scaled the box and the number of worms, then not. But if I took bigger boxes and put the same number of worms, then maybe yes, because they would have more space and their tunnels might not interact the much they do in this box.
So you would get the different results with different size of the box?
I assume yes.
Okay. Do you think that the parameters like type of the soil you're using could affect the result? I mean, I don't know anything about it, so.
I think yes, but clay was an optimal choice because it is very hard and the worms actually have to do some work to it. While if I choose some ground that was already porous, it wouldn't have much effect on it.
Mm-hmm. Did you measure the porosity at the beginning of the experiment also?
No, I didn't, but the porosity the porosity of the box with no worms, I will be comparing that to the other ones.
Okay. Okay, could you go to the last slide? What is this slide showing?
It is showing the results of the percentage of water in the soil. So in the first box, on the number 1, these are three different measurements from different places in the first box that I took and gave to the researchers, which helped me. And they measured the average, which was 22.8%.
So somebody measured that for you?
Yes.
Okay, and what would mean 100% of the water in the soil?
That would mean that there is no soil at all.
Okay, that would mean that you have water.
Yes.
And just one question for the opponent. In your presentation, you said that the name and the anatomy of the worm is a problem. Why do you think that is?
No, I actually said that it's very well that it's satisfactory how he put the picture of the anatomy of the worm into his presentation.
But why is that so?
Well, it explains a bit why how the processing of the soil is done, but I think he should have explained a little bit more with words and what each part of the body and organ does, how it
Okay, but that's for the anatomy, but what about the name? Is the name of the worm telling you something?
The name. Well, I like how he investigated a bit about the species of worms.
Okay, I mean, good thing would be that if you want to repeat the experiment, then you know what worms he used, and then you can use the same worm. Yes, that would be the good.
That's good. Yes,
Okay, that's all from me.
Okay, I would like to go from the reviewer to the reporter. The opponent, can you show us your presentation, please?
Okay.
Last slides. Please, last slide, just.
With what?
Last slide.
Okay, so
Just go to it.
Go down.
Go down.
okay, so this one.
Okay, reviewer, what were the conclusions of these points? So, what is the conclusion of the point of conditions in the second experiment?
Conditions in second experiment. I don't understand.
What was their conclusion? What did the opponent and the reporter conclude? Okay, grow to the porosity. What do they conclude about that?
If we have more worms, the porosity will grow.
Did the opponent had the same thinking about that?
I think yes.
Okay, do you agree with them?
Why?
Yes.
Yes.
Okay, so my point is that you should go through the points for discussion. You should say what did the reporter said, what did the opponent said, and what is your opinion. Yes. Also try to use your time for clarifying questions so you understand the bits of their presentations, or if you are not sure what did the opponent maybe think that the reporter's opinion of the growth is true, then you should ask him about that. Okay, so for opponent, in your template when it's written the problem statement, you should really copy-paste the problem statement if you can.
Okay.
Now it's okay, but in the future. Can you tell me what experiments that he did? The first experiment was about what, and the second?
The first experiment
What he wanted to investigate?
in the last term.
In first. What he wanted to
in the previous term.
investigate?
Okay.
Yes.
If I remember correctly, it was the dendrochronology.
No, in this problem,
What?
in this
I
problem,
don't-
the first experiment, he had two experiments.
Oh, the first
What he wanted to investigate?
The first experiment, he wanted to investigate how the porosity of the soil depends on the number of worms.
Okay, and the second experiment?
And in the second experiment, I don't remember correctly.
Okay, you should be aware of what experiments your reporter did. Okay, so for the reporter, first question is, okay, what was your second experiment?
My second experiment was that I am currently doing it. I planted 100 seeds and I will be measuring.
But no, the second experiment which you had results. You showed the results of second experiment, if I'm not mistaken.
No, that is the experiment
That's the second. Okay.
with porosity. I measured the porosity and the moisture at the same time.
Okay, how would the porosity change over time?
Over time, well, the worms will be digging more and more tunnels, but at the same time they would actually be collapsing other tunnels. So I think that 30 days, 28 days was enough to measure the porosity at its maximum.
Okay, you should investigate that.
Yes.
Maybe. Why do you think that 28 days is enough?
Well, that's a month. That's enough time to give worms to work on the soil to make it more porous. But as you see, the measurements takes long, so
the best thing would be to investigate how the porosity changes over time and to know that 28 days is okay or not. Maybe you should use a shorter time. You could see it from that experiment. On the graph you had 3 columns for every box.
yes.
You should use one column, and you had error bar. So and also maybe try to zoom, not to limit your y-axis, not to start from 0 but from some number, so the difference can be seen.
Okay.
So we can compare what was the error on
Yeah,
every.
but the results were so similar. There were
Okay, but
mistakes
on
in measurement were
my
wrong.
computer, for the first 3, I see a small difference. And I don't know, okay, that's from 20 to 25%, on 0 to 30% showed, and how much is that, I don't know.
Yeah.
You should write down next to the graph or something like that.
Okay, well, the differences were in 0.01%. So that is almost nothing.
Okay, but you should state it because
It's not seen in the graph yet.
yes. Also, so what are quantitative parameters that you have investigated?
So I investigated the amount of worms that were placed. I investigated how moisture affects, how worms affect moisture, porosity, and growth of plants.
Okay, you should have maybe some slide that you state what were the parameters that you are going to investigate, and in conclusion you should try to say how, and maybe if you could, okay, you have here 3 points for every moisture level, but maybe try to have more if it's possible.
Yeah, but the measurement
Just to see if it's, linear or exponential or something.
Okay.
Okay, you said that the porosity will change linearly or exponentially in one case. Can you draw that and explain why you think that should be?
Okay. So let's say this is the rate of porosity, and this is box number 1 with no worms, 2 with 25, and 3 with 50 worms. I believe that it would be either something like this or something more like this.
Sorry, what's on the y-axis?
Rate of porosity.
Rate of porosity, and you are increasing the number of worms on x-axis.
Yes, 0, 25, 50.
And you think exponential would be like that? Okay,
Yes.
but do you think in ecosystem, if you have more of the same animals, what would happen to them? So if they have the same amount of food and you have more of them, what will happen?
It would probably...
And also the porosity cannot escape the 100%.
Yes.
So I'm not. I don't agree with you that it would be exponentially in growth exponentially or linearly, because
Okay.
you need to have what is the edge case. What if we
Okay,
have
so you think it would stagnate at some point of worms?
probably,
Yeah.
but I'm not sure it would stagnate or fall some, but it would have some limit.
Yes, of course, if I put like 1,000 worms in,
Yes.
then it would fall to 0, but
Would-
that's not natural. So I try to replicate natural systems, and this would be optimal for them.
Okay, you said that you were using the worms from your region.
Yes.
Why didn't you use the soil from your region?
I did use the soil actually
Okay.
from the same region.
Okay, then tell that.
Okay.
Because the worms are accustomed to that soil. Also, when I think Goran asked you, why didn't you use, or the opponent I think that asked you, why did you use the soil just from, and okay.
Does the
never
type
mind.
of the soil affect the experiment?
I think that maybe you said that the porosity on the start would be the same as in the end, because there is porosity.
No, it's
in the
not the
beginning.
same at the start as in the end, because we moisturize, so
Okay.
moisture rearranges the particles of the soil.
The question was asking if he measured the porosity at the beginning of the experiment.
Yeah.
Oh, okay. It would be interesting, or would you know what would happen if you pour low concentration concrete into the soil and remove the earth around it? Would it show you something maybe?
Well, maybe it would show the tunnels of the worms, but I don't know how it would play out because it could actually destroy the tunnels, its weight could destroy the tunnels and make the ground denser. So that would not be completely correct measuring to do.
The reason why you use low concentration. But you could try it just to see what happens. Also, when you don't isolate wood, you should maybe try to measure and the soil percentage of water in soil when you after some time after you put the water in the soil.
Okay, so in different times after I put the water.
Yes,
Okay.
just to know what's the difference when the water is in the whole soil and the wood also.
Okay.
Yes. You didn't conclude your report. You were
Yeah,
on.
because I had no other measurements than that. The conclusion was on the graph.
In future, you should
Okay.
have some. And the goal would be to make a numerical model or just part of the model so you can quantify what is the influence of every. Maybe try to narrow down the problem and try to investigate quantitative parameters. So, you can now see that it's exponential for this parameter is linear, and so on. Okay, Thomas, do you have questions?
I
Go ahead.
have just one. Did you check if the worms were alive at the end of the experiment?
Yes, they were all alive.
Okay.
How did you count them?
Yes, I counted them.
How did you count them?
I took them out of the ground. I put the ground on the floor and I just counted them from each box.
So they were all like 50
Yes.
and 100. Maybe it would be useful to have this graph, this number of worms, because people forget what was box number 2, what was box number 3. Maybe you can just put number of worms.
Okay.
No worms, 50 worms. If you can just comment on your graph, last graph. The difference between box 2 and box 1 was larger than the difference between box 3 and box 2. Do you have any explanation for that?
I believe it was pretty similarly different, but maybe it was a little bit more between box 1 and 2 because in the first one there is no worms at all. So that's quite a bit difference because there is actual some activity in the second one and in the third one, but in the first one there is no activity at all, just water and ground.
Is it the difference, like the number of tunnels that 50 worms make during the time of the experiment could be supposed to be the same as number of like 50 worms in the third box are making in that time? So I would expect some linear relationship. Maybe it's not, but this suggests something that is growing faster. We don't know if it's true because we have just a very limited number of measurements. But if it is true, what could be the reason for that?
The reason for exponential number of
We don't know if it's exponential, it might be quadratic. I mean, just 3 points. We cannot decide on this trend, is it linear or whatever, but what might be the reason for non-linear relationship? Maybe you have an idea, maybe you don't, it's fine if you don't.
Yeah, because some worms cross other tunnels of worms and they collapse or something happens to them when I pour the water in, that might be the cause of it.
But then you would expect less in...
Yes, but...
and we see more of this moisture.
Maybe because 50 worms decrease more slime than the 25. And they increase slime, so it actually adds up to the water, to the moisture, right? So maybe that is the reason, but I'm not certain at all.
Okay, just rhetorical question. Why did you choose a method that you cannot carry out yourself? Because you are now dependent on somebody else.
Yeah, this is the most correct way to approach this problem because it is precise, it is done by professionals, it has to be correct. I mean, the formula is, well, the formula that is used is correct, so I believe that this is the best way to approach this problem.
Maybe you should investigate how they do it and try to put the method. Just one slide to explain why,
Yeah, they will
and
do
why
that.
did you, use this cut? Why did you use it?
Because in the vacuum desiccator all moisture is removed, so all moisture will be can be counted. That's
Okay, you should maybe have a picture of that instrument and explain what happens.
Yeah, I haven't got, but I will get all the results, all the ways they measured it, and all the pictures of the instruments.
Because the goal of the tournament is you should ask for help, but you should try to do it with them, or